What's up, guys? Welcome to PhD Biology. This video is going to serve as part 2 into our study into DNA. So to finish off this big topic, we're going to talk about transcription and translation, the way our cells use our DNA to produce proteins that we need. So with that said, let's get started. So to start us off, we need to make a clear distinction. DNA is genetic material. It controls the traits that we see in each generation. But it does not directly produce any proteins, even though it certainly has code for those proteins. To get proteins, we begin with this process called transcription in the nucleus of the cell. The ultimate goal of transcription is going to be transcribing codes in our DNA into messenger RNA, better known as mRNA. We think of DNA as the template for this mRNA. So to define transcription, we'll say that transcription is the synthesis of mRNA under the direction of DNA. So our first step in transcription is going to be called initiation. During initiation, an enzyme called RNA polymerase II, which you can see right here, this purple, the enzyme is going to attach to something called a promoter region in our DNA. The promoter region is actually not shown in this picture, but it would be back here before anything even starts. So promoter region is just a site in our DNA that shows the RNA polymerase where to begin. Promoter regions contain what we call a Tata box, which again is not shown, but it's an area of DNA where you have thymine and adenine occurring repetitively, hence the TATA pattern, so we call that a Tata box. So that's in the promoter region, and to attach to the promoter region, RNA polymerase actually needs some help, so we have these helper proteins called transcription factors, which basically show this enzyme where our Tata box or the promoter region is to get it started. Now we get into the second step of transcription called elongation. So before we start, notice that when the RNA polymerase is building mRNA, we're only doing it on one strand of DNA. This strand of DNA is not being used. We call the strand that we use the antisense strand. That's a good little thing to know for AP and IB. The antisense strand is where we make the mRNA. So anyway, in elongation, the enzyme moves along the template or antisense strand in the 3', which would be right here, and 5', which would be right here. So it moves in a 3' to 5' direction, adding complementary RNA nucleotides as it goes. And remember that complementary RNA nucleotides are just like DNA, except instead of thymine, we're going to have uracil. After this, we hit the final step, which is termination. Termination just means that the RNA polymerase reaches a certain termination site where it's told to stop building this mRNA. So we then have mRNA that can be set free from the DNA. After transcription, we need to process the mRNA before translation. So we go through something called RNA processing. This process is only going to occur in eukaryotic cells. Keep in mind that prokaryotes are going to skip this step. Anyway, during RNA processing, we edit the mRNA. The 5' end, which would be this end or this end, is the 5' end. It gets a guanine cap. You can see they label it cap. It gets a guanine cap that's going to protect the RNA. And the 3' end is going to get a poly A tail, which just helps the mRNA move through the cytoplasm. We also cut out these sections of the mRNA called introns. We're going to cut them out. Introns are just non-coding areas of mRNA, meaning that they serve no purpose for later coding. What we keep are these called exons. So the introns are spliced out by a cool protein called a spliceosome. So it cuts out introns and leaves the exons to form our final mRNA. After all this has happened, once we get this nice little mRNA, we can move on to translation. So for translation, you're going to need to understand how to use this table. So I'll explain that in a minute, but it's crucial that you know this for AP and IB exams. So when translation begins, the mRNA is now in the cytoplasm and it attaches to a ribosome to begin protein synthesis. So to make a protein, as we learned in the biochem video, we need a specific order of amino acids. Now to get this, if we look over here for a second, we're going to take the nucleotides on our mRNA strand and break them up into groups of 3. And we're going to call these groups of 3 codons. So if you would look, this is group 3, AUG. This would be a group of 3, UGG, and so forth. So each codon calls for a different amino acid. There's always going to be a start codon, coded as AUG, which calls for the amino acid methionine, and that tells the protein to begin the building process. We also get certain stop codons. If you look, UAA is a stop, UAG is a stop, UGA is a stop. That stops the process of building proteins and stops bringing in more amino acids. So let's talk about the way we use this table before we explain the actual process. So the way we use this table is to find out which amino acid a codon is calling for. This table is always used for codons, what we see in the mRNA. So the way we do it is like this. Let's say we have a codon ACU, adenine, cytosine, and uracil. So we look, the first letter is an A, so we look in this row. Then the second letter in ACU is C, so we're going to look in this box because the row and the column meet in this box. And then we're going to have a U, so we can look here, and the U is this one. So we see that we have ACU, which codes for something, which codes for an amino acid called threonine. So that's how you use these tables to figure out which amino acids come for any given codon. Now let's talk about the specific steps in translation. Translation is the synthesis of a polypeptide by a ribosome under the direction of mRNA. So as I mentioned, protein synthesis happens at a ribosome. This big thing is a ribosome. So in the ribosome, we have an A site, we have a P site, and we have an E site. The way this works is this. At the A site, right here, the codon, or set of 3 nucleotides, is red. So right now at the A site is UGG. So when it's red, we get transfer RNA, or tRNA, to find the anticodon to pair with it. An anticodon is just the complementary base pairs for the codon. So if the codon is UGG, the anticodon is ACC. Also, tRNA carries a specific amino acid, as you can see. And we figure out which one by using, again, this fun table. So after the tRNA enters at the A site and kind of attaches, it moves over to the P site. As you can see, we have methionine at the P site. And when it moves to the P site, another tRNA enters the A site. Once you have something in the P and A site, the amino acid in the P site is going to attach to the amino acid in the A site, and it's going to detach from the transfer RNA. So you have you have a tRNA in the A site with an amino acid on top, and imagine this would be connected to the amino acid. Then just this tRNA can move over to the E site, where it leaves the ribosome. This process continues until a codon in the mRNA has one of these stop codes. So when we get a stop code, this is going to stop. And after this happens, translation is complete. And we get a chain of amino acids known as a polypeptide or a protein. That's going to be about it for our lesson into DNA and its functions. If this video was helpful, leave a like. As always, if this video is kind of confusing, you can comment with any additional questions you may have. And don't forget to subscribe to see more biology videos. Thanks for watching.